This is the next video of the chapter cell, and this is the other organelle after the endomembranous system, that is ribosomes. After microbodies, it comes after microbodies. Ribosomes are also known as pallet particles. This is the only organelle which has organelle in itself. In 70s type of ribosomes, there are organelles. Okay, and ribosome itself is an organelle. So it is the only organelle that also has an organelle in itself. It is present in all living cells, both prokaryote and eukaryote. It is also called as ribonucleoprotein bodies because they are consisted of RNA and protein that we will learn in the structure later on. And these are the smallest membraneless organelles. They don't have any numel or matrix. They only have RNA and proteins. And proteins are the ribosomal proteins. These are also known as protein factory as they are the site of the protein synthesis. Now their structure consists of two subunits which are small and large. Both the subunits are present in cytoplasm separately. However, they come together at the time of protein synthesis, and that also in the presence of magnesium ions to form what is known as ribosome. Okay. This small subunit is cap-like shaped, and on its lower end it has a cleft or depression-like structure that is actually a hollow space. And the largest subunit has a groove and has a crab-like structure that can hold the above small subunit. Okay. Um, at time of protein synthesis, the small subunit joins the large subunit so that the cleft fits above the prototuberous. And what are prototuberances? These are those trap-like structures which are three in number and present in the largest subunit. And this is also that part that contains the groove from where the polypeptide arises. We had learnt about this as we were doing earlier in the endomembranous system. We were learning about ribofourin. Yes, we have done this. Okay. Now measurement of ribosome is usually done by sedimentation coefficient, which is also known as Swedberg's unit, and this is actually the rate of sedimentation, and it is a measurement that indicates actually the density and the mass of the ribosomes. And also the letter capital S does not denote Swedberg's units. Okay, Swedberg units is just a name so that the scientist could be privileged by because of his discovery, but actually the capital S is used for sedimentation coefficient. It is the correct name. Swedberg's unit is not the correct name. Now types of ribosome. Prokaryotes and eukaryotes. Prokaryotics have the 70s ribosomes, and eukaryotic cells have 80s ribosomes. Prokaryotic have subunits 30, 50, and subunits of eukaryote are 40, 60. Prokaryotes are located in the cytoplasm of the prokaryotic cell, and that of the eukaryotic cell are present in the cytoplasm of eukaryotic cell. Now they may be present in either freely in the cytoplasm or maybe attached to the plasma membrane or cell membrane. Okay, now in prokaryote attached they are if they are attached to the cell membrane, then 50s subunit of them is attached, and okay, free maybe 50, maybe 30. And inside eukaryotic organelles such as mitochondria and chloroplast as well, prokaryotic cells are okay, prokaryotic ribosomes are also present. But in eukaryotic cells, no prokaryote ribosome, that is 70s ribosome, can be found anywhere except mitochondria and chloroplast. Eukaryotic cells have prokaryotic ribosomes, but prokaryotic cells don't have the eukaryotic ribosomes. That is it. Yes. And now in the eukaryotic ribosomes, that is ATS ribosomes, if they are attached to the membrane of the RER or rough endoplasmic membrane, we have learned that in the function of reticulum. Okay, and also present in the nuclear envelope, and here it is 60 years that is present. Okay, both the large units of the Both the prokaryotic and eukaryotic ribosomes are present in the cell membrane, membranous structures or envelopes or okay. Then composition of ribosomes. Ribosomes, as I told, are only made up of RNA. Okay, that is ribosomal RNA and R proteins. It is also single stranded, and there is a fact that has to be learned that rRNA is the most stable type of RNA present. Okay, R stands for ribosomal here. Now ribosomes have subunits 30S, 50S for 70S, and 80S has 40S and 60S. Now RNA, that is the constitute of rRNA and proteins. Now rRNA of 30S subunit has 16S RNA, and protein is they have 21 proteins. Similarly, 50S of 70S ribosomes has 23S of RNA and 5S of RNA as well, and they have 33 proteins. Similarly, 80S of 40S of 80S has 18S and 34 proteins, and 60S contains 28S, 5.8S and 5S, and has 40 to 45 proteins. Whole table is to be redundant. Okay, now 70S का 30 to 23S as well as the 28S of 80S. Now these two are actually catalytic in nature, and that is because of the enzyme present in them, that is ribozyme, and these are the enzymes that help in the peptidal transferase. Okay, these two enzymes 32S and 28S. Now we had also learned about the peptide polypeptide formation, and the peptide bond is formed between two amylose acids to form a polypeptide bond. Now the two amino acids, that is, see, this is an amino acid. Okay, again this is an amino acid, this is an amino acid, and this continues on, and the polypeptide bond formed between them is formed between these two amino acid. Okay, this is a peptide bond, polypeptide bond, and it is formed between two amino acids because of this peptidal transferase enzyme. And this is peptide transferase are the only enzyme in nature that can form polypeptide, and nowhere else this bond between these amino acids can be formed by any other enzyme, only and only by peptidal transferase. Okay, now a ribosome is also itself considered as a catalytic organelle just because it has catalytic enzyme peptidal transferase. Wrote learned. Okay, biogenesis of ribosome, that is synthesis of ribosomes, usually occurs in the nucleus in the eukaryotic and the cytoplasm of prokaryotes. Now, mitochondria, the other organelle, it is also known as the powerhouse of the cell because it has ATP synthesis for all the cellular process. The synthesis here is done by the oxidative phosphorylation. The energy obtained is by the oxidation of glucose. Okay, oxidative phosphorylation, that is how the synthesis is done. Now discovery was done by Kolliker. He had observed the mitochondria for the first time, and if you try to go to the first or third page of the copy, you will find at Altman had gave the name bioplast, and Fleming gave the name phyla, and Benda actually then gave the name of mitochondria. Earlier it was given two names, bioplast by Altman, okay, phyla by Fleming. Mesozymes have been called chondroids also. Okay, another information to be remembered. Mesosomes have also been called chondroids. Now, staining of mitochondria is done by Janus green. It is a vital dye, and it is a unique stain for only and only mitochondria. Now, if we apply Janus green, that is a vital dye, to a living cell, then the mitochondria present in the living cell will change its color to blue. Why? Because of the enzyme cytochrome oxidase present in the mitochondria. Okay, now it is a marker in enzyme of the mitochondria. Marker meaning only and only found in mitochondria, as a ribosome had only and only peptidal transferase was the only enzyme to form polypeptide. Similarly, okay, the marker enzyme is only and only found in mitochondria, and it is an oxidative enzyme. Okay, if the cell is living, cytochrome oxidase will oxidize the Janus green to blue, and if the dead, then no color change because of this cytochrome oxidase. Okay, now the number of mitochondria varies. An animal cell have more mitochondria than plants because definitely plants have chloroplast for their cellular photosynthesis respiration. Okay, and also because they have a very big vacuole, and flight muscles of birds, that is how the wings muscles, okay, from with the fly, can have about 50,000 to 5 lakh mitochondria. That is for the quick energy of for flight. Now structure of mitochondria, it is a double membrane organelle. First point. Now then there is outer membrane, outer chamber, inner chamber, and cistern. What? Outer membrane chamber, inner membrane chamber, and cistern. Now it is a double membrane organelle. First, outer membrane. The outer membrane is smooth, and the lipid protein ratio here is 40:60. And also the outer membrane has porins. We had read this also in the first class. Okay, and porins are for transport across the membrane. Now outer chamber that comes after outer membrane also has perimitochondrial space. This is the space which it includes the chamber. Okay, it is not single membrane, it is actually a chamber, so it is a space, and this space is also called perimitochondrial space. Now it is poor enzymes, that means it won't be functioning very good. Okay, since it is poor enzymes, so common sense, it won't be performing much of that biochemical reactions and other things. Now inner membrane is physiologically active, therefore it has various infolds that are known as cristae. Okay, cristae membrane has a lipid protein ratio of 20:80. It has cardiolipids. What are cardiolipids? They are the modified lipids for attaching special electron carriers. An electron carriers such as cytochrome and ubiquinone are very popular. Are the electrons carriers such as cytochrome and ubiquinone. Remember these two names. Hmm. Next, cisternae also has F0 F1 particles or oxiosomes. Okay, oxiosomes F0 F1 particles. Sorry, cristae has following enzymes: ATP synthetase and succinate dehydrogenate. And in the inner membrane, that is the inner chamber, that is the last that we have to study, has ground chamber or matrix, and this matrix have many things like enzymes for the process of Krebs cycle, hydrogen or protein carriers like NAD plus and FAD plus, mineral ions like ferric and cuprous and manganese. Okay, now ADP and inorganic phosphate also are present here, and it also has the circular DNA, DS circular DNA, also has some 70s ribosomes present in them, and a polysome complex as well. Here is the diagram of mitochondria. You can see this is outer membrane, outer chamber, this area, and outer membrane is a lipid pressure 60 to 40, also has porins. Outer chamber is less, okay, less working because it has less enzymes. Now 70 S ribosomes are present inside the matrix, and inner membrane, then inner chamber. Okay, F0 F1 particles along the cristae itself. Okay, and then again DS circular DNA and the polysome complex. Now cristae, if we highly magnify cristae, okay, this is the diagram of cristae. It should be very well in your mind. Now this is the F1 head, okay, of the cristae, and it is extrinsic protein in actually, or enzyme. It is or enzyme ATP synthetase. Now it may be in extrinsic protein, or it may be enzyme ATP synthesis. Now F naught is the base, that is intrinsic protein, and it is permeable to proton, therefore is also known as tunnel protein, tunnel proton. Okay, It is also called as proton tunnel. So it helps in the movement of protons intrinsic, since it is intrinsic, and F1, that is head, is extrinsic, and so it is called F0 F1. Okay, and then here we have protons. Now the concentration of the protons outside the crystal and inside the crystal are to be different. Okay, inside the proton has to be high in large amount, and outside it has to be of low low density. Okay, now